Hello everyone, I'm Isaac and I'm a junior research fellow in medical sciences and also a cell biologist at the Dunn School of Pathology. Wadham is a really special place for cell biologists because Robert Hooke was a research assistant here, and shortly after he left the college, he invented one of the first compound microscope, and using which he observed an array of tiny boxes on the cork, and he called them cells because it reminded him of the life of the monks in the monastery, and because they live in the cells. And again, a few hundred years later, here we are. We have a more powerful microscope, and that allows us to go deeper into the finer structures of the cells. And today we're going to talk a little bit about the world of organelles. So organelle, as its name implies, they are the organs of the cells performing vital functions. So I will give you a few examples. In this book, in this page, you can see here is a fluorescent image, and the green line marks the boundary of a single cell, and the white circle right in the middle is called nucleus, and it's an organelle where the genetic material called DNA are stored. And there are a lot of red dots scattering across the whole cells, and they're called mitochondria. And you can consider them like a battery or powerhouse of the cell to generate energy enough for the functions of the cells. So you may wonder at this point, you can just pass around maybe to have a look. At this point, why certain organelles, they are huge, like the nucleus, and why other certain organelles, they are quite small, like the mitochondria? What is the rules governing their size? And today I'm going to talk about a few theories about the organelle size control. But before I start, I'd like to introduce another organelle called the centriole. And it's a very important organelle in our body, and it's almost inside every single cell in our body. And they're important because the organelle, this centriole, will organize the pericentriolar material, and then to form the spindle that pull the chromosome apart into the daughter cells. And for the male, in sperm, they also form a very long tails that allows the sperm to swim and for the continuity of the humanity. And also within our trachea, our breathing tracts, they form the cilia that will sweep the mucus and try to remove the mucus from our breathing tract. So therefore, they're really important. But despite of its importance, they are really simple. So you can see I brought a 3D printed model of centriole, and it has a perfect 9-fold rotational symmetry and as a cross section, and it also has a length. So the organelle size can be essentially reduced to a 1D problem. You just measure the size of the centriole, and this gives you a proxy of the size of the organelle. So we try to use the centriole here as a model to understand how the cell regulates the organelle size. And you can just pass around and have a feel about, and it's really inside every one of us. So there have been a few theories about how centriole regulate its size, why the centriole only grow to approximately 200 nanometer, but not too long, not too short. What would be the mechanism behind that? So I'm going to tell you 3 different ideas, and the first one is called a limited components hypothesis. So this idea postulates that the cell will produce the exact numbers of the building blocks required to build the centriole. Let's say the centriole is right here, only 5 building blocks in length. So right like here, so it's 5 building blocks in length and with 5 units of the building blocks. It means that the cell will actually produce exactly 5 copies of the building blocks. And they will find each other inside the cell in order to assemble into the organelle of 5 units in length. So what do you think? So what would be the mechanism that allows us to test this idea? So we can make use of a technology called the green fluorescent proteins. And I actually bring one. It's a green fluorescent protein from my lab, and also a protein that without any fluorescence, so it's transparent. And you can pass around again just to have a feel about it. And when the protein attacks with the green fluorescent protein, it becomes visible. So I use a sticker here to represent a green fluorescent protein for each building block that is tagged with a green fluorescent protein. Therefore, we can think about that if the cell produces 5 units of the green fluorescent building block, And then we can actually see them in the cytoplasm, like this. So if they find each other, and then they form the centriole, and you will also assemble into an organelle with exactly 5 fluorescent units. So that's the beauty of the microscope that allows us you to see the signals. So this is the first idea. And the second idea is called a molecular ruler hypothesis. So it postulates that there is rulers in the cell. So what it means is, so that if there's a rulers, and the cell, if they want to assemble into an organelle, so you will keep adding the building blocks to the cell until it reaches the end of the ruler. So let's say, for example, this block is actually like taller than the rulers. Then you just be discarded. So the cell will build exactly to the length of this molecular, build this organelle with exactly to the length of this molecular rulers. And this is the molecular, the rulers hypothesis. And the experiment that we do is, first of all, we have to identify what is the ruler that is governing the size of the cell. And this is the hardest part, because sometimes you may not be able to identify anything. But if you could identify a molecular rulers, what you can do is you can genetically change the length of the rulers. So let's say, for example, right now, we can just make it longer. Then our idea is that if the ruler is longer, then the centriole will be able to grow to a longer size. And right now, if we tag with the fluorescent tag, then we will be able to look at it with a more fluorescent signal into the organelle. So conversely, if you make the molecular ruler much smaller, and then it would just build a much shorter centriole. And I'm going to tell you one of the last hypotheses is called dynamic balance hypothesis. So this idea is that the size of the organelle will be controlled by exactly how much the building blocks is adding to the centriole versus how much the building blocks is leaving the centrioles. So it's a dynamic balance between something adding in and something is removing. So the idea is very simple. Let's say, for example, at each unit of time, I'm going to add 2 building blocks to the centriole, and then I'm going to take away 1. So at another time point, I'm going to add 2, and then I'm going to take away 1. And then you can see that it's actually growing in the net centriole 1 unit at a time, although we're adding 2 and we take away 1. The third time point, take away 2, and then I add 2 and take away 1. And then the last time point, add 2 and take away 1. So right now the centriole has grown to 4 units of the building blocks. So when we end, it ends when the centriole adding in, the building block adding into the centriole will be exactly the building blocks leaving the centriole. So we reach an equilibrium that of the 6 units. So it's keep adding in and keep being removed. So how can we test this hypothesis? Is we make use of a technique called the FRAP, the fluorescent recovery after photobleaching. So the idea is that we shine an extremely strong laser to the centriole. What it essentially do is it will switch off the green fluorescent protein. So right now all the block become transparent and without any color, like this. So what it suggests that if it is in a dynamic balance with the surrounding, and if it keep exchanging, then some of the fluorescing molecules in the surrounding will be adding into the system. So right now we start to see some of the green signal from the centriole. And then, because it's a dynamic balance, it's also going to remove some and then adding back some. So over time, because the surrounding with so many building blocks compared to the centriole, then eventually we would expect that it will recover back to its original signal. So that is why we can use this technique to tell whether it's in using this kind of model to control the organelle size. So today I'm just briefly talk about the 3 different mechanisms in which cells can use to regulate their organelle size. But in reality, they're really complicated. They possibly use a mixture of all these strategies, or perhaps, as you know, the cells, they are the best engineer. They can invent their own way and new ways that beyond our imagination to build their central size. And so that's all, and I'm happy to take any question, and thank you for your attention.